a regulation in at the interface between translation and growth control. So I think that the recorded version just missed that introduction of you, Carson. So I will repeat that you're an associate professor in the Department of Molecular and Cellular Physiology at Yale. And then I said more wonderful things about you, but you can keep going now. You can get started. Thanks.
All right. Thank you, Karla. That's a very nice introduction. Let me get this set up. Okay. All right. Am I good? Cool. Well,
You're
gosh,
good.
Stefania's talk is a hard talk to follow. But hopefully you will be excited to hear about some of the RNA related experiments we've been doing. So here we go. So one of the main questions in my lab is to try to understand what are all of the different regulatory sequences that can be encoded in mRNAs that make them do interesting things, especially post-transcriptionally. And I think one of the main things we've learned over the last 30 years or so is that basically every single feature of an mRNA has regulatory potential. And this is just showing a map of a selection of them. But everything from the cap structure to sequences encoded in UTRs, codon choice, 3' UTR sequences or poly A tail can affect the post-transcriptional fate of mRNAs. And we'd like to, there's still a lot that we don't understand. And so there's a certain amount that you can get from just studying endogenous mRNAs, but at some point, a more powerful approach is to use massively parallel reporter assays. And so basically the way these work is that you can synthesize thousands of different mRNA fragments, and then you stick them in a reporter mRNA and see what happens in your assay. And so this approach has been used to identify regulatory RNA sequences in 5' UTRs or in coding sequence or in 3' UTRs. And I think it's been really powerful in helping us understand which sequences control things like translation or stability. So one part of the mRNA that this hasn't been used to interrogate are the very 5' terminal sequences adjacent to the cap. And the reason is really just that promoters that are primarily used in these plasmid-based assays add on a fixed 5' UTR sequence. And so even though these RNAs may have different 5' UTRs, the very 5' terminal sequence is often constant. So why might this 5' terminal sequence be interesting? Well, one main reason is that that's the location of the cap. And the cap, as I think probably most of you know, is chemical structure that's added on to the RNA co-transcriptionally. And throughout the course of the life cycle of the mRNA is bound by a broad series of different key regulatory proteins. So as soon as the mRNA is transcribed and capped, it's bound by nuclear export factors that drag it out of the nucleus and into the cytoplasm. And once it's in the cytoplasm, it can be recognized by translation factors, but also different translation repressors. And then during the decay process, a key step, of course, is chopping off the cap. And so all of these factors primarily recognize the cap structure itself, but are bound directly next to 5' terminal sequences. And so I think it's not a stretch to imagine that those sequences in certain cases might influence the activity of some of these different processes. So even so, there's really only one well-established example of an important regulatory 5' terminal sequence, and it's called a terminal oligopyrimidine motif, or a top motif. And so what this motif is it starts with a C, and a key feature is that the C really needs to be in the +1 position. If you insert even a single non-C nucleotide in front of it, completely undo the function of this motif. And then it's followed by between 4 and 14 pyrimidines. And this is not a very common motif. It's found mostly on ribosomal protein mRNAs and other translation factors, so probably between 200 to 300 different mRNAs. And the key functional piece of this sequence is that sticking it onto the end of an mRNA makes that RNA translationally hypersensitive to nutrient signals that are transmitted through the TOR pathway. So when the TOR pathway is active, these RNAs are highly translated. But under conditions where the TOR pathway is inactive, these mRNAs become acutely and rapidly translationally repressed. So are there any other 5' terminal sequences other than those classical top motifs? So one reason to think that there might be are that 5' sequence usage is fairly conserved across species. And so what I'm showing you here are frequencies of transcription start sites. And just as a side note, I'm going to use transcription start sites in 5' terminal sequences interchangeably. One refers to the sequence in DNA, the other, of course, refers to the RNA sequence. So these are TSS sequences, TSS fibers used in HeLa cells, compared to TSS frequencies in zebrafish that are assayed by the same method. And what you can see is that there's a really striking correlation. And this correlation exists with Drosophila, though the strength of the correlation is a little bit reduced, but also as evolutionarily distant as Arabidopsis. So I think what this says to me, at least, is that the selection of different 5' terminal sequences is really quite well conserved. Interestingly, this degree of conservation falls apart once you compare human sequences to yeast, indicating that this is likely a metazoan phenomenon. Okay, so how can we test the functions of different 5' sequences? Well, to do this, we used really a very simple, straightforward, massively parallel reporter approach. So the main innovation here is on our plasmid, we used a CMV promoter. And amongst the commonly used promoters for ectopic expression, CMV promoter is really quite unique in that it starts transcription at preferentially a single position. And it starts transcription there almost no matter what sequence you give it. And this is really ideal for us where we want to test all possible different 5' sequences. And so after that site, we included a random set of 7 nucleotides, and then a 5' UTR, and then ranello-luciferase. So then we put this plasmid library into cells, made RNA, and we can measure the frequencies of different sequences by reverse transcribing and then analyzing the library using next generation sequencing. So here's an example of some of the sequences, just to show you that we get transcription initiating at the site we expect. So this is the set of 7 random nucleotides, and this is the very end of the CMV promoter. And you can see that transcription really robustly starts preferentially at this +1 nucleotide. So I do want to use this opportunity, though, to tell you about one caveat that is frankly a little bit embarrassing. But the strategy we use to prepare these libraries means that we can't reliably quantify the frequency of +1 G sequences. So what I'm going to show you throughout the rest of the talk only includes +1 A, C, and U sequences. So the first thing we did was just check how well all of these different sequences are expressed. And we were able to detect about 99% of the 16,000+ sequences in the library. But what we noticed was this very strange bimodal distribution of expression. And we were puzzled by this for about a few minutes until we started looking at which sequences were at the top and bottom. And this revealed that really +1 nucleotides were the primary determinant of the level of expression. So what I'm showing you here in red are all of the +1 A sequences that were, more or less uniformly pretty well expressed. So conversely, RNAs that initiated a +1 T are quite poorly expressed, while +1 C mRNAs span this whole range. So we also tested whether or not there were any differences in stability, and they're not. So those differences in expression levels really reflect differences in transcription. So are there different patterns between these classes of mRNAs? So if you look at nucleotide frequencies in the top expressed +1A mRNAs, you find not anything really that striking. There's a slight depletion of A nucleotides at the +2 position, but otherwise fairly good representation of different nucleotides. For +1C sequences, there's really quite a striking difference. So this again are nucleotide frequencies in the top 5 expressed +1C sequences, and nearly all of them have a T nucleotide at the +2 position of the 5' end. Past that, there's a slight depletion of A's and a slight enrichment of C's. So the +1T TSSs are similar in some ways to the +1Cs, except they don't have this strong enrichment for T's in the +2 position. So in some ways, these results are not totally unexpected. So the motifs for the +1A TSSs look quite a lot like the canonical initiator motif. So the initiator motif is defined here with the A being the +1 nucleotide. And this is really present at about 50% of transcription start sites in mammalian promoters. And really for our purposes, the only notable feature is a lack of an A at the +2. We don't see a total absence of an A at the +2, but there is a depletion. For +1C RNAs, this pattern looks very much like what's known as a TCT motif. And this is a unique promoter motif that's primarily found on ribosomal protein mRNAs. And again, you can see this motif was defined as having a strong requirement for a T in the +2 position. So this said to us that at least, our artificial promoter system is recapitulating some known transcription motifs. So this is, of course, though, an artificial system. So we also wanted to know how well do these TSS frequencies actually compare to what occurs in cells? So this is mapping the TSS frequency of different trimers in our library versus TSS frequencies in endogenous HeLa cell, RNAs in HeLa cells. And what you see is that there's quite a strong correlation. And nicely, +1A mRNAs are uniformly pretty well expressed. +1T or uRNAs are pretty poorly expressed. And +1C mRNAs are sort of in the middle, with the best performing ones being CT sequences. So the transcription rules that I think come out of this are that +1A mRNAs are pretty uniformly well expressed and don't really have too many sequence requirements. +1C RNAs can also be expressed almost as well as +1A RNAs, though having a T in the +2 position is of great benefit. Whereas if you want to make a +1T or +1U RNA, it's all tiers. So of course, the thing that we were really interested in is using this system to determine whether other 5' sequences had post-transcriptional functions. And the first thing we decided to test is whether we could identify any sequences with translation functions. And the reason we suspected that we might find something is that the EIF translation factor, which is one of the key translation factors in the initiation process, recognizes the mRNA cap and makes interactions with the 5' terminal sequence. So each of these different components of EIF4F can be cross-linked to the first 12 nucleotides of mRNAs, suggesting that the identity of those nucleotides might somehow impact its function. So to test this, the approach we did was just to put our library back into cells and then run cell extracts over sucrose gradients to isolate polysome-associated RNA and subpolysome-associated RNA. And when we analyzed these results, they were actually quite disappointing. We found that there really weren't any striking translation differences between specific sequences in our library. So you can see here that +1A RNAs are a little bit more polysome-associated, +1C RNAs are a little bit less polysome-associated, as are +1U RNAs. But there was no obvious motif other than the first nucleotide identity that correlated with differences in polysome association. So then we started to think, well, maybe under conditions where EIF4F is active, 4F can bind essentially every mRNA somewhat equally well, and that masks a lot of the differences of 5' sequence function. So what happens when EIF4F is inhibited? So I mentioned this briefly at the beginning, but the primary regulator of this translation factor is the mTOR complex 1 pathway, which senses nutrient signals. And if we turn off mTOR complex 1, either by nutrient deprivation or inhibitors, like this mTOR inhibitor, TORN1, 4F is disassembled and dissociated from RNAs. And this exposes its 5' end so that it can now be bound by other cap binding proteins. So one example of this is a protein called LARP1. So LARP1 is the translation repressor of top RNAs, and the way that it works is whenever 4F is displaced, LARP1 can come along and bind RNAs that have both a cap and a top sequence. And LARP1 will come up later on. So to measure translation functions of 5' sequences under conditions where cap-dependent translation is inhibited, we again ran cell extracts over sucrose gradients after treating those cells with the mTOR inhibitor TORN1. And you can see, as expected, a strong depletion of polysomes and a strong increase in subpolysomal ribosomes. And when we looked at sequences, the changes in the translation of individual 5' sequences, we saw a really striking difference. So now all of the distributions are substantially more spread out. +1a mRNAs are relatively a little bit less repressed, whereas +1c mRNAs as a group are much more repressed, and the distribution is much, much broader. And so when we looked at the very far end of this distribution, what we find sort of expectedly is that the strongest repressed sequences look like classical top sequences. So +1c followed by a series of pyrimidines. So we wondered if we can infer something more about what defines top motifs and the strength of their repression. So the biggest thing that varies with top sequences is their length. So we bend RNAs based on the length of a top sequence. So what you can see is if you have a sequence that's just purines and add single C, there's not really any change in translation. If you add another pyrimidine, again, there's not much of an effect. But now if you add 3 pyrimidines and then 4 all the way up to 5, there's sort of a nice linear increase in the degree of repression. And this was sort of gratifying because if you look at a crystal structure, and this is a crystal structure solved by Andrea Berman's lab, showing the top binding domain of LARP1, what you find is that it accommodates exactly 5 pyrimidine nucleotides. So there's at 3 pyrimidines, you see a little bit of repression, but at 5, you get maximal repression, probably by increasing affinity based on these interactions. So we also wondered whether we could infer functions of individual positions of top sequences. And so to do this, what we did is we started off with sort of a canonical top sequence set of a +1c and then 4 pyrimidines. And we tested what would happen if we changed each position to each other nucleotide. So what you can see here is if instead of a C, we put an A or a T, we almost completely undo the translation sensitivity of those sequences. If we make those same changes at the +2 position, however, it's much less sensitive. This is a little bit puzzling, but the +2 position, at least for translation regulation, does not seem all that important. The +3 position, on the other hand, is quite sensitive. Putting a G in the +3 position also almost completely undoes the regulatory potential. And regulatory significance decreases as you get further and further from the cap. So in some ways, I'm sure some of you will recognize that these sequence patterns look a little bit like the TCT patterns. And so we also wondered how these changes affect expression. So again, to get maximal expression, really, you would have to put an A at the +1 position, although this would completely negate translation regulatory function. So a C is sort of a nice compromise. And then at the +2 position, in sort of contrary to what happens with translation regulation, there's a really significant requirement for a plus 2T for maximal expression. And then the impact of these nucleotides decreases, of course, as you get further away from the +1 position. And so what we concluded here was that the first 3 nucleotides are really the key determinants of both expression level and translation regulation. And that those motifs for both translation and expression are similar, but not quite the same. So another way to look at this relationship is just to plot all of these 3 mers with, on one axis, expression level, and on the y-axis, change in translation. And I think what this shows is a really interesting sorting of different 3-mer sequences. So all of the +1A sequences sort of cluster together, with the exception of AAA, which we don't fully understand. But these are pretty uniformly well expressed and not that translationally regulated. All the +1T sequences are quite lowly expressed and also not that translationally regulated, where +1Cs span this whole region. And what's interesting is down in this corner is a set of four TSS sequences that have both pretty good expression and pretty strong translation regulation. And if you look at TSSs that are used across endogenous mRNAs, so this is TSS frequencies across some cell lines and other human tissues, what you find are those exact 4 sequences are also preferentially used in endogenous mRNAs. So one exception that you might see is this CTA sequence in liver samples. And it turns out that that's an exception that sort of proves the rule, because those sequences are almost fully accounted for by the massive expression of albumin. So these patterns are also conserved across species. So again, this is looking at TSS frequencies between humans and mice, and this is across all mRNAs. And what you can see is that those 4 +1C sequences are really preferentially used. If you look at ribosomal protein mRNAs, which I told you previously, like to use TCT promoter motifs and also code-related top motifs, these are really the top used TSS sequences. And if you look at zebrafish and Drosophila, you see those patterns recapitulated. So especially in ribosomal protein mRNAs, once again, those 4 3-mers are the most heavily used in ribosomal protein mRNAs. And again, in yeast, this is quite different. So for yeast rpMRNAs and for yeast RNAs in general, +1C sequences are really not used nearly at all. So the final thing that I wanted to come back to are differences in stability. So I told you at the beginning that under control conditions, these library mRNAs are all similarly stable, and there's not much interesting to see. But because inhibiting the TOR pathway affected translation so much, we wondered whether it would also affect stability. And indeed it does, not as dramatically as it does translational control, but what you can see is that the distribution both broadens, and there's a slight stabilization of +1C mRNAs and a slight destabilization of +1A mRNAs. So to show you what this looks like for some individual transcripts, we made 2 +1C RNAs and 2 +1A RNAs. You can see under control conditions, these are in a docs-off reporter, so we can shut off transcription and then follow the decay over time. So under control conditions, these are more or less similarly stable. But if we inhibit the TOR pathway, what we see is a really nice and quite dramatic separation. And I just want to remind you that these are all mRNAs that only differ in the first 7 nucleotides and are otherwise identical. So a final thing that I just want to add that we think is interesting that we don't fully understand is the relationship between stability and translation. So again, under control conditions, there is nothing really that interesting to look at. But under conditions where we inhibit the TOR pathway, what we see is a really quite striking inverse relationship between the stability of mRNAs and their change in translation. So for +1C mRNAs, sort of as we expect, they are both translationally repressed, but also somewhat stabilized. And we think this probably reflects action of LARP1 and indicates that LARP1 is likely a regulator of many +1C mRNAs beyond just classical top mRNAs. But what we don't understand is the nature of this relationship for +1A and +1U mRNAs. So if anybody has any great ideas, please let me know. So I'm going to end there. The conclusions that I hope you'll remember from this talk are that TSSs define unique transcription and post-transcriptional profiles, and that different TSS use has widely different effects on mRNA fate, that many endogenous TSSs reflect hybrids of regulatory motifs that impact both transcription and post-transcriptional functions, and that 5' sequences can link the translation and stability of mRNAs. So this work was done primarily by Tony Vanden Elzen, a former postdoc in the lab, and also with a lot of help from Megan Watson. And I will end there.
Okay, Carson, great. Turn your video on so we can talk to you. That was really wonderful, detailed foray into translation regulation. I'm just looking at the Q&A, and I think people must still be typing. I wanted to ask you about 2' O methylation because you've been dealing solely with nucleotide identity, and I wondered if somehow the nucleotide identities near the TSS, the fact that they tend to get 2' O methylated at the first and second position could somehow play into your assay.
Yeah, I think it's a really interesting idea that we haven't tested. I think all mRNAs have methylation at the +1 position, and about half of mRNAs have methylation at the +2 position. And I'm not sure that there's a clear sense yet of what the sequence determinants, or if there are sequence determinants that affect that. So I guess we could repeat this assay with deletions of different methyltransferases, but we haven't done that yet. But I think it's an interesting idea.
I mean, one thing that seems obvious is the first nucleotide will tend not to be in structures, or tend to be less in structures than the second nucleotide, maybe, if you just think about it like probabilities, folding probabilities.
Yeah, that could be definitely hairpins that bury the +1 nucleotide are really repressive translationally.
But you haven't done anything sort of folding prediction wise with your sequence hits.
No.
No. Yeah. Okay, well, we're not, come on guys, type faster. We're at 5 o'clock, so there's a bunch of people still here, so they obviously want to talk to you, but they're shy or something. So look, 2 new messages. Yes, please submit your questions on the speaker Q&A. There's still quite a lot of people here. Come on. We'll just wait a few more minutes and then. What's the next experiment?
Oh, geez.
You didn't have to answer that. You're probably tired.
That's a tough question.
I thought this was amazing that you got this homologies between species in part of your motivation. And also the other kind of amazement came from another side, which was just from our work on like long read sequencing, where we seem to see a lot of fraying at the TSS, where we've talked about this before, but I wondered if you wanted to comment on this, because it seems to us that, and maybe this is some kind of artifact of our methods, but we see a lot of variability within a few nucleotides around TSSs. Is your method somehow more accurate and you really are primarily getting one nucleotide for the start?
So I don't think so. I mean, I think that, when when I'm showing TSS frequencies, it's including promoters that start at a sort of dispersed set of sites, right? So I think what you see is pretty common, where transcription doesn't start at a single site, starts at a range of sites in a region.
Okay, so you're counting each read independently.
Yep.
That's
Yep.
where your scores come from. I love that. Okay, good. Here's a question from Layla. Thank you. What would you predict would happen for G as the first nucleotide?
Yeah, so the problem with detecting Gs in our system is that MMLV reverse transcriptases tend to add a C when they reverse transcribe that's non-templated. And that's a random process, and it's different from batch to batch. And so what happens is we get a lot of reads that started at the +2 position, but then they have a non-templated G edit. So that's why we can't quantify them. But if we sort of do some filtering that gets rid of a lot of the artifacts, the +1 G RNAs look a lot like the +1 A RNAs. And so our expectation is that those sequences are pretty similar.
Gotcha. Okay. Well, there's still 30 people there. I don't know what they're doing. They're pondering, hey, Stefania is coming back. Great. Let's have a martini.
No, I mean, can we log off or we have to be here?
No, you're right. We can log off. I'm just sad. These were two great talks. I really enjoyed them. Thank you.
Just so everybody knows, our next talk is on March 29th. It's hosted by Alberta RNA Research and Training Institute. And we can't wait to see you there. Thank you all for coming out, and thank you to Carson and Stefania and Karla. You all were amazing. So
okay, thank you.
We're all clapping.
Thanks, Karla.
Bye.